Hello, I'm Andrew Skinn. I'm the founder of the Ruskinn company, now known as Baker Ruskinn. And I'd like to just take a few moments to talk about the journey that my company and the subsequent products have gone on to bring what we have today, which is the best incubation and culture technology for stem cells into Malaysia in this partnership with Yakin. So my company was founded 30 years ago and has always been involved in microbiology, and from 1999 got involved in the whole field of cell biology and understanding how cells behave when they're in the physiological environment of the body. And that journey really started with the University of Oxford in the UK, with a team of very pioneering scientists there that were interested in the impact of oxygen on cell physiology, particularly in the field of kidney and cancer at that time. Long story short, that 20-year journey led to Sir Peter Radcliffe and his laboratory at Oxford, and Sir Peter Radcliffe being awarded the Nobel Prize for his discoveries in how oxygen signals in the cellular system, a thing called hypoxia inducible factor. Now, applying that across all different fields of science, of course, we moved to therapeutics, and we moved to areas like IVF and all sorts of mechanisms which involve taking cells from the body and then doing a manipulation outside the body and then returning to that environment. And so that's kind of IVF and stem cell therapy, and Yakin are really trailblazing the way forward in terms of stem cell therapy, because what we have seen in the team here is their desire to really do things in the best way possible. And the best way possible means that the engine of their machine needs to be delivering the best quality of biological quality of stem cells. So all our learnings and all the science that Ruskinn and Baker Ruskinn have brought to this team really are to demonstrate the importance of continuing the physiological journey that cells see from being removed from a body, being manipulated and grown in a laboratory, and then injected back. So what we have as a pioneering venture here with Baker Ruskinn and Yakin is the world's first stem cell production facility that is done under mimicked physiological conditions. And by that, we are not talking about the normal process of laboratory with incubators. We're talking about an isolator within which the environment is at 5% oxygen and mimicking the humidity and temperature seen in the body. So those cells derived from umbilical cord are really on a journey that totally mimics the natural environment. And of course, what we see is that will lead to the best efficacy when it comes to implantation in the patient. So this is a really interesting project, and Yakin are the first. They have the first isolation system in their Yakin Medic factory here, and Baker Ruskinn are really keen to work with this team to develop the technology and the solution as a new therapy for Southeast Asia. And what we bring to that team is our expertise, our technology, of course, but also our connection to the scientific networks around the world. I've spent my life traveling the world, connecting with the leading scientists, understanding what the issues are that they face, and those issues require technical solutions. So I've been very proud to be alongside that journey of pioneering new technology that delivers future science, that improves the quality of the scientific outcome and improves relevance of that scientific outcome to real therapies. So we're bringing that network with Dr. Chris Duranton and I to Yakin and to this venture here, and it's an exciting time, and we're very much looking forward to seeing the impact and the results of our collaboration.
Hi, I'm Dr. Chris Duranton, and I'm the Director of Scientific Applications at Baker Ruskinn. With Yakin, we're working towards creating novel stem cell-based therapies for regenerative medicine. So my role here as a scientist with a background in oxygen biology is to make us work with correct oxygen concentrations when we work with stem cells. Now, this is really important because when we talk about stem cells and the origin of stem cells in the body, we have to understand that these so-called stem cell niches are hypoxic or physoxic, which means that they are in physiological oxygen concentration. Now, most of you know that the atmospheric oxygen concentration is somewhere around 21% of oxygen. Now, this is not true in the human body, where cells and tissues reside somewhere in between 3 to 13% of oxygen. Now, this is because oxygen doesn't readily travel through tissues and liquid, so the diffusion of oxygen distance is very short. Therefore, stem cells too originate from physoxic conditions. Now, in order for the stem cells and exosomes, for that matter, to be effective in treatment, we have to make sure that the entire journey of the cell from the body to the recipient, so patient to patient, is kept physiological. Oxygen is a very potent molecule. It creates reactive oxygen species very easily, which damage biomolecules. So we, together with Yakin, have focused on creating a physoxic path directly from the donor and where we collect the stem cells and exosomes throughout the journey, which is kept physiological, all the way to the recipient, the patient. In this way, we can ensure that the cells we use are of high quality and they perform as stem cells should, as in being able to deliver and develop into any cell type. Now, we work hard. We have a lot of scientists working with this, and I'm so very proud to be one of them. Now, we focus on delivering the best tools for regenerative medicine in this very novel field of stem cells and exosomes, but we know that with the backup of our scientific community, we will deliver the best and most efficient treatments.
Hi, Assalamualaikum. My name is Dr. Fazlina from UKM. I would like to thank the organizer for inviting us today to listen to very impressive talk by this speaker. And for today's seminar, we learned a lot about the culture system that can actually help us to improve the way that we do research in lab. And I really enjoy a lot of things, idea that can actually improve our research setting in the lab. Thank you very much.
Hi, I'm Dr. Angela Ng from the Center of Tissue Engineering and Regenerative Medicine at the University Kebangsaan Malaysia. I would like to thank Yakin Splendour for the invitation and Dr. Krista from Baker Ruskinn for the great talk. She highlighted the importance of physiological condition, maintaining a physiological condition for cell culture, and the concept of physioxic in cell culture. With that, maybe we could harness the best therapeutic effect of the cells that we culture, for example, MSCs. With that, I would like to thank Yakin Splendour and Baker Ruskinn for bringing this platform to Malaysia, and we hope Baker Ruskinn will continue in its R&D and continue to develop its solution for the betterment of science. Thank you.
Hi, my name is Dr. Shariza Nordin. I'm from the University of Putra, Malaysia. I wish to take this opportunity to thank Yakin Splendour for inviting me to attend this talk. It's very nice, very good, very informative, and I wish all the best to everyone. Thank you.
Hi, I'm Dr. Yogesh from Center for Tissue Engineering and Regenerative Medicine, UKM. I'm also currently serve as the President for Tissue Engineering and Regenerative Society of Malaysia, TESMA. So it was an interesting talk organized by Yakin Splendour and provided by Dr. Krista from Baker Ruskinn, Finland. So it opened up a new paradigm on hypoxic cultured MSC derived exosome. So far we have discussed a lot, argued on the hypoxic cultured MSC compared to the normoxic MSCs. So now this provided a lot of insight and info on the exosome, the importance of exosome culture in MSC in hypoxic condition. So I hope the Baker Ruskinn can provide more data, studies, and also equipments for other researchers to expand their studies on hypoxic condition derived exosomes to expand this study on this area.
Hi everyone, I'm Daniel from Center of Tissue Engineering and Regenerative Medicine, UKM. So I'm currently working as a senior lecturer there. So today's talk is by Dr. Krista from Baker Ruskinn. It's very interesting, especially because it's something I'm working at right now as well. So actually, the exosome is something new in the field, and extracellular vesicle have received a lot of attention recently. So I'm also working with EV derived from mesenchymal stem cells. However, today's talk is inspiring because I haven't yet worked with the hypoxic MSC culture hypoxic MSC derived EV. So it's actually hopefully in future, we can have a potentially collaborate with Yakin and Baker Ruskinn to work and study more about the hypoxic MSC derived EV. Thank you.
Hi, I'm Sarah, a PhD student from UPM. So today I attended a talk by Dr. Krista on the topic of hypoxia exosomes, organized by Yakin Splendour. So the talk is very interesting and insightful because it is very close related to my project, which is on exosomes. So I would like to thank Yakin Splendour for organizing such events, and I am keen to involve more on this kind of events in the future. Thank you.